We're going to keep persevering here, and we have Brad Hine from CSIRO who's going to present here. Mark is going to present on pain and wellbeing to the audience in Adelaide. In terms of for us that aren't here today, that will be recorded and you'll be able to view that, so don't cry just yet. And we've got Brad here today to talk about immune competence, which is a very interesting subject, and some pretty cool stuff's been done in this space. So Brad is a research scientist with CSIRO. He's based just out the highway at Chiswick. His expertise is in ruminant immunology and leads several projects around this, aiming to develop methodologies to assess it in both sheep and cattle, and developing alternatives to antibiotics for use in food-producing animals to prevent and treat disease. Brad has particular interest in this resilience area and assessing it, and is related to their ability to cope with challenges they face in the production environment and investigating how these traits impact on health, welfare and productivity in a changing production environment. So ladies and gentlemen, put your hands together today for Brad Hine and his talk on improving the resilience of livestock.
So thanks for the introduction, Luke, and I just wanted to thank Sheep Genetics and MLA for the invite to talk to you today. So as Luke mentioned, I've got a particular interest in looking at resilience in livestock, and that's what the talk will be about today, is strategies to help farmers improve resilience. So with the recent floods, you would have heard the word resilience or resilient people a lot in the news recently. And resilience is used in a lot of different ways in different contexts, and I thought I probably should give you my view on what I think resilience means in livestock as a starting point. So for me, resilience is really about the ability of an animal to cope with challenges they face in their production environment and return to being productive again. So we know that animals in our production systems face a whole range of environmental challenges, and these are just a few. They're many and varied. So they face climatic extremes, and that's becoming more and more the case, as we know, with climate change impacts. There's also stress induced by management practices. We don't intend to induce stress in animals, but we do through marking and weaning. They're necessary things in the production system. There's also challenges from disease-causing organisms, or what I'll call pathogens, and you'll hear me mention the word pathogens quite a few times today. So that's what I'm talking about, any type of infectious agent that can cause disease. But we know that sheep and cattle are also very social animals, and they get used to their structure in the herd, or their position in the herd or the flock. And when we disrupt that, it can be quite stressful for animals. And a good example of that is that social mixing of cattle at feedlot induction is a high risk factor for bovine respiratory disease. So I guess what's important to know is that animals respond to these various challenges with a series of host defence reactions, we call them. And we won't go into too much detail today, but just for you to understand, they're varied. So there's physiological responses, there's immunological responses and behavioural responses. And the more work I do in this area, we actually see that all of these responses are really integrated with each other. So each one influences the other. And I think it's really important that we look at all of them together. It's very dangerous to look at one on its own because they all have impacts on each other. So if you're an Angus stud stock producer and you've got a nice paddock, nice group of bulls in your paddock, and I said to you, can you predict which of those bulls is likely to have progeny with the best growth potential? You could probably do a pretty good job at that by looking at EBVs for growth. So you've got the tools and the information to help you as a producer to answer that question. Similarly for marbling potential. But if I ask you to tell me which of those bulls in your paddock are likely to have progeny with the most robust immune system, best ability to fight disease, that's a much more difficult question for producers to answer. Similar with heat tolerance, and you heard Hamish talk recently about some of the priorities around heat tolerance and selecting animals that can tolerate heat, and that's becoming more and more important with a changing climate. The other one that's really important is ability to cope with stress. We all cope with stress differently, and so do animals. Some animals can handle stress better than others, and it can impact on lots of things. So the bottom three there all sort of feed into an animal's resilience phenotype, but the problem is at the moment we don't have a lot of information about those particular traits. So why bother improving resilience? Why is it important? And I think the big issue is that we're asking animals to be productive in an ever-changing production environment. So we know climatic extremes, we see that all the time. The other big one is emerging diseases. We've all just seen what COVID's done to the world, okay? It's an emerging disease. These types of diseases, it's very hard to vaccinate against something you don't know about. And you've seen that in the news with the COVID. It takes time to develop vaccines to protect animals or people against diseases we don't yet know about. So that just highlights the importance of strategies around improving the resilience of animals to cope with those challenges, some which we don't know about yet. And also increasing intensification. So the livestock industries are trying to feed a hungry world with protein, and as part of that, intensification of livestock production systems is increasing, and with that comes more challenges. And I guess as producers, the take-home message for you today is that as we ask animals to be more and more productive, we are actually reducing their ability to cope with environmental challenges. Now that's really well documented when you look across lots of food-producing species, that as we push the animals harder and harder from a production sense, we reduce their ability to cope with challenges. And that's a really important message for producers going forward. So why don't we currently select for resilience traits? One of the big things I think, and it's an easy perception to have, is that the most productive animal is the most resilient one. Otherwise, how did they get there in the first place? But we know that that's not actually the case. So if you look, for example, in the dairy industry is a good example where mastitis and milk production are the incidence of mastitis and productivity are highly correlated. So as you push animals harder to be productive, incidence of mastitis goes up. And that's the same situation for many common diseases we see in livestock and pigs and poultry, that production traits are unfavourably correlated with disease incidence. So the other one is a really obvious one. They're really hard to measure. So something like weaning weight, pretty easy to measure weaning weight. We run the weaners over the scales when we wean them, we've got the trait nailed. So there's lots of data on weaning weight. When you're asking someone to look to define a phenotype of how strong an animal's immune system is, it's a lot more difficult, costly and time-consuming to do. So as a consequence of these things, there are limited tools for producers, and that's what our work is really all about, is trying to put tools in producers' hands to help them start to add some selection emphasis on the resilience moving forward. So key part of resilience is an animal's ability to resist disease. Now, I'm not claiming it's the only one, it's a very important one, but there's a whole lot of other factors that contribute to an animal's resilience, but it is a key one, obviously, is their ability to resist disease. So we know immune responses in livestock are heritable, and we've known that for a long time. So their ability to respond to a challenge has a heritable component. So that's good news, we can start to select for it. And we've been very successful in the livestock industry for many, many years now in breeding for resistance to specific diseases. Parasites, internal parasites in sheep, there's lots of different examples. Brucellosis in cattle, there's lots and lots. We've been very good at it for a very long time. But what I'll talk to you more about today is this idea of breeding for general disease resistance, and that's what I call immune competence. So immune competence is about measuring the strength of an animal's immune system to handle a variety of disease challenges, not just one challenge in particular. And I'll explain to you a little bit more why that's important. So this was really a concept that Bonnie Mallard and Bruce Wilkie over at the University of Guelph in Canada first sort of come up with early in the 80s and 90s, and it's really their work in pigs and dairy cattle that have pushed this general concept of immune competence down the track. So we're sort of riding on their coattails a bit. So I was fortunate enough to spend some time in Bonnie's lab working with dairy cattle and then come back to Australia to try and apply those same concepts and principles into Australian production systems. So we won't go into an immunology lesson today, except to just say that our immune system is very smart. If it wasn't, we wouldn't be here, because we're always fighting bugs that are also very smart. So our immune system tailors how it responds to a pathogen so that whatever response we mount is the most effective response to try and eliminate that threat. So this is a real generalisation of how you respond to a challenge, but we can largely split how you respond into type 1 and type 2 responses. So type 1 responses, which are cell-mediated responses, and I always describe this to people as, you probably remember the old Pac-Man game, that the Pac-Man would come and gobble things up. Well, in a really basic sense, that's what cell-mediated immunity is about. It's really effective at things that go inside your cells, so things like viruses. Cell-mediated immunity is really important for that. Pac-Man comes along, gobbles up those infected cells and eliminates them. But we also, the one we hear most about, of course, is antibodies. So we all know when we go and get vaccinated, when you get a tetanus shot or whatever it happens to be, your body produces antibodies to whatever the target was, and they're the things that hang around in your system and protect you when you see that same thing 6 months or 6 years down the track. Now, they're really effective at controlling things that are outside the cells, but they can't get inside the cells. So that's just a really broad difference. So the message here is that there's two types of responses in general that we mount, and it's important if you want to have general disease resistance that you're pretty good at both. The other thing it tells us is if you select for resistance to a specific disease that relies on one arm of the immune system, sometimes that can actually increase your susceptibility to diseases that require the other arm of the immune system. So animals that are good at one type are not necessarily good at the other. And that's a really important message. So we certainly don't advocate to stop breeding for resistance to specific diseases that are economically important to your industry, but we say do it with caution and tag along with breeding for general disease resistance at the same time, so that you don't push your animals down one track. So the general idea, it's a pretty simple protocol, and it needs to be if it's practical to apply on farm. So when we started to think about how we might do this, it was the first priority for us was it had to be practical to apply on farm, and had to not restrict the future sale of animals for producers. So we started at the end and worked backwards. That makes it a little bit harder for us to do the things we want to do, but it's got to be practical for industry to take it up. So what we do is we vaccinate animals at the day of weaning and ask them to respond to that vaccination. Why we do it on the day of weaning is because livestock are just like us. When they get stressed, they get sick. And we know in our production systems that at our peak times when we get high disease incidents, and they generally occur around stressful periods. Weaning, induction to the feedlot, all of these things where the animals are under some stress impacts on the ability of the immune system to respond. So what we're trying to do here is ask an animal to respond when they're under stress, because we're interested in those animals that can do that rather than respond to a challenge when they're out with mum and they're happy and healthy out in the paddock just walking around. So we want to look at those animals when you put them under some stress can still mount a really strong response. We measure the immune response to that vaccination, and it's a clostridial vaccination. There's no secrets here, there's no rocket science here at all. And then, but the key part of what we do is we combine measures of cell-mediated immunity and antibody-mediated immunity, and that's how we rank animals. So we want the animals that are good at both. If you're good at one and you're crappy at the other, they're not the ones we want. We want those animals that have got a good balanced response and can do both. Then we come up with this immunocompetence phenotype, and what we're working on and when we'll have to continue to work on is to show that that actually relates to general disease resistance. So we then thought about what sort of animals did we want to look at? And we're very fortunate that MLA funded 2 projects early on here, one in beef cattle and one in sheep, and this was done on the resource flock animals, the sheep part that Hamish has just spoken about. And it was really important for us to use the right group of animals, and that's where these resource flocks are such a valuable asset for research. So I can't speak highly enough about that, because what they provide is it's a setting where we can do our research, but it's industry relevant because they run under commercial conditions, and that's really important. There's extensive phenotypic data collected, which we could never collect ourselves, and we're really interested in what happens if you select for immune competence or resilience to all of those other traits we're interested in. We can't look at that on our own, so resource flocks provide the opportunity for us to look at our component and start to investigate those associations. It's important when we're testing across multiple locations, and we need to know that this works across multiple locations, that there's link size. And the really important one for us is that the animals are genotyped, and that's because we've always looked at working towards developing a genomic test for this. These traits that are hard to measure, expensive to measure and time consuming, genomic tests, are the end game to try and help take that out into industry. So the aims of those initial projects were really to develop methodologies to assess immune competence in sheep and cattle, and as I said before, the important criteria here was that they were practical to apply on farm. We need to be able to go onto a farm, do lots of animals quickly, get them out of the yards, and get them back on feed. So that's really important. We wanted to look at genetic parameters around the immune competence traits. We wanted to see how heritable the traits were, to see what the potential was to select for those traits. And as I mentioned, we're really interested in relationships between immune competence and those other resilience traits like stress coping ability and heat tolerance, those kind of things, and also important production traits. And of course, the end game here is to determine the ability of our immune competence phenotype to predict health outcomes, and that's what we're trying to do now. We're not at the end game by a long way, but we're making progress along the way, and I'll talk more about an MLA-funded project through the livestock consortium, which is allowing us to further validate that work. So this is just a little snapshot of the benchmarking calves. So we work very closely with Angus Australia and have done since the very start of this work, maybe in 2013. So in the early phases where we were developing the methods, we were assessing their benchmarking calves, and this was cohort 2 and 3. We're now testing cohort 10, so that's how long we've been doing this for. And you'll see here that we tested 1,149 animals, various locations, so we're testing their immune competence phenotype on farm, following them through the feedlot and looking at their health outcomes at the feedlot. Now this is a reasonably small data set in the scheme of things, but it's a good starting point, so about 1,000 animals. So a couple of things that were really interesting to us, and hopefully will be interesting to you, is just the variation in responses that you see in your herd. So on the bottom axis, the number refers to a herd, and the letter refers to a year drop. And you'll see generally across the years, they'll track together. So the top graph here is looking at weaning weight, and this is the kind of variability we see in weaning weight in a group of calves. Now, we take that a little step further. This is within a contemporary group. So these calves have been raised together from birth, and they're AI calves, so there's not a lot of split in age, from birth through to weaning weight. So there are a couple of weeks variation in age, but that's the kind of difference you see in weaning weight. This is the kind of difference you see in cell-mediated response. It's enormous. Now, these animals have had the same immunological history, I say, because they've been raised in the same contemporary group. So we can't say for sure, but from the best of our ability, they've been exposed to the same kind of disease challenges on the road to getting there. And we see the same thing in antibody response. So when you vaccinate your animals and you think they all respond the same, you're very mistaken. You'll get animals... These are actually antibody responses to tetanus in your 7-in-1 vaccine or your 5-in-1 vaccine. We'll get animals that we hardly see a response at all through to animals that'll blow it off the chart, and we see everything in between. And we know a real challenge in livestock health are those animals that don't respond or are low responders to vaccination. They're very hard to protect because they just don't mount a response that is protective when they see that pathogen again. Oh, I should go back. The other thing we saw was that the traits were moderately heritable, so around 25 to 35%. I think the antibody was around 30% and the cell-mediated was a little fraction higher. So that was encouraging. Of course, there's only 1,000 animals there, so pretty big errors with those estimates at that point. But we have confirmed that with a lot more animals now, and that they were pretty close to where we thought they were. So then we followed these animals through the feedlot and looked at their health. So you'll see what we did is we took the extremes of immune competence phenotype. So low animals, pretty ordinary at antibody production, pretty ordinary at cell-mediated. They're the animals we expected to have the most issues with disease. The high ones are those that are right up the top. So they're really good for antibody, really good for cell. And then there's a whole swag in the middle that we just bunched together. So you look at reported health issues, it followed the trend we were hoping. For every 100 head, 15 reported health issues, only 10 in the average high, so that was good. But what we didn't expect was the sick days. So when we talk about sick days, this is at the feedlot. So an animal's sick, they're pulled from their feedlot pen, they go to a hospital pen, and those sick days are whatever comes first. It's either from when they were pulled from the pen to they succumb to the disease and are no longer with us, or they return to the pen and be productive again. And you'll see that this increased with the immune competence phenotype. We've also looked at this data as a continuum as well, and we see exactly the same sort of pattern. So of course we get the thumbs down there, but hopefully the next slide will explain why. So what was interesting is that we had about 6% mortalities in those low animals. We had none in our highs and about 1% in the average animals. So what was happening is those high animals, they still get sick, but they recover and go back to being productive again. And that was what was increasing their sick days. It was because they weren't dying. They were getting better and going back to the pens. So of course these are low numbers. I mean, we've only got 1,000 animals here, so we can't get too excited about these results. We need to validate this further and further before we can be confident, but they're at least encouraging to us from that early work. So what we were able to do is get data from the feedlot on the cost of the animals, how much the drugs cost, what it costs to feed them if they prior to them dying from disease, all of those different things, and come up with a cost per head due to health-related matters at the feedlot. And what you can see is for the high animals, it was about $3.50 a head. For low animals, about $103 a head. So this was quite interesting to the feedlotters, but what was more interesting to them is it was only 11% of the animals we said were low responders, but they contributed to 35% of their health costs. So if you can get those 11% out of the system, there's a big saving for you as a feedlot operator. So current and future projects. So I mentioned that we're fortunate that MLA funded a validation project around this immune competence work as part of the National Livestock Genetics Consortium. And this is really about trying to further validate those findings. We acknowledge we've only done 1,000 animals at that stage. We need to go out and do more animals to actually validate that our immune competence phenotype predicts health outcomes reasonably accurately. So this was all about using commercial animals. So the benchmarking calves are terrific animals to work on to do that genotype work and get all the phenotypes, but they're run at a gold standard. So they're all pre-vaccinated to BRD before they get to the feedlot. They're not mixed at the feedlot. Everything's done to the letter with those calves. So we wanted to look at animals that are just what normally is going through commercial feedlots in Australia. So what we've done, we've tested 1,600 calves on commercial farms at weaning, and that was in the middle of the drought, so that was a bit of a challenge. And it'll be interesting to see how we go here because they were very small. These are animals that would normally be 250 kilo, we'd be testing were probably 120 kilo when we tested them, and these guys were doing it tough. So they could not have been under any more stress when we tested their immune response, so that'll be interesting to see how all plays out. So we're in the process now. They're all but one small group are at the feedlot, and the first group has come out the other end. So we've got health data on them, and we also follow them through to slaughter and look at offal at slaughter to pick up any health issues we might have missed. So any of those subclinical health issues along the way that we haven't picked up. So that's ongoing, and in the next couple of years we'll have the results on that. And it's really important that we continue to do this, because we have to demonstrate to producers that we can predict health outcomes, and that's where we're at. Until we can do that, we can't tell you to select for immune competence till we've got the data to back it up. So the other work we've been doing, as I mentioned, we continue to work with Angus Australia with their benchmarking calves. We're up to cohort 10 now, and they fund us every year to go and immune competence test another group of calves. And so we do around 1,000 calves a year. We've done around 5,000 calves now, so we're starting to get a reasonable data set together for the geneticists to do their number crunching with. So the real goals of this work with Angus Australia are twofold. One is a genomic test for commercial beef producers. So some of you will already know Angus Australia already have a Heifer Select product out there, which is around genomic tests for selection of replacement females in commercial herds. And we're also currently working with Angus Australia to develop a similar product for steers called Steer Select, which is all about predicting feedlot performance in commercial steers. So this will hopefully be released in the next few months. We've done a lot of validation work there. But what we're hoping is, as we get more and more information to say that selecting for immune competence is a good thing, that it will be incorporated into these genomic tests to allow commercial producers to start to get some information on those. So some of you may have seen in the last year or so some information coming out about what we've called Immunodex, and they're research breeding values that we've generated with Angus Australia on their benchmarking sires. So these are obviously for those seed stock producers to start to select for immune competence, but they're just research breeding values at this stage till we get more evidence that we're on the right track. So they're just a couple of projects that are ongoing currently. Now, geneticists, please shut your eyes when you look at this slide, because the errors are fairly large, shall I say, but that's okay. So this is some work we've been doing that's funded by AWI as part of the MLP project that's run out at Chiswick. And this is an add-on project where we've taken the weather progeny from that project and we've immune competence tested those weathers. As part of this project, we've done about 1,500 lambs that we've phenotyped, and then 50% of them were retained for lifetime measurements similar to what happens with the ewes, so essentially retained as wool growers, and the other half were finished for a commercial lamb feedlot and on to slaughter. But what we did do on the way was keep very, very detailed health records on all of these animals. Now, because there's not a lot of them, these correlations with immune competence have very large errors. So the results are encouraging, but we've got to do a lot more work here, is what that's telling us. And the only way to get these errors down is to do more and more animals. But it was encouraging. So on the left-hand side, this is breech strike, post-weaning weck, hogget weck. This is yearling fleece rot, post-weaning dag. And the bottom two, these are what we call fitness compromise indexes that we've made up. So the illness one, any illness that a lamb has had in its life will attract a score, and this is a cumulative score of any health issues that animal has had through its life. Things that kill an animal will receive the highest score. Things that give them a little bit of a setback will give them a low score, but they accumulate. And this one's a total fitness compromise, so it includes lots of things like congenital issues, anything that has compromised the fitness of that animal in their life. So the encouraging thing, if you see a smiley face, it means the relationship was the way we wanted it to be. So for breech strike, it was negative, meaning a high immune competent lamb that we tested had a lower incidence of breech strike. And you can see that they're all negative, which is what we'd hoped for, except fleece rot. And I must admit, because this was done largely in the drought, we didn't have a lot of fleece rot data. So hopefully that proves to not be, that association is not that way in the end. But as I emphasise, these have got very large errors, these estimates, so we can't really take much from them at this stage, apart from saying it's encouraging. We need to get out there and do a whole stack more animals to try and get those errors down and find out what the real values are there. So that's about it from me. So I guess where we're heading is we would like to develop a similar index in sheep as we have in cattle. And we've just come up with this name Residex, whether that's what it really is or not. But what it's about, Residex, for me in the sheep game, it's about an index that helps producers maximise management efficiency while optimising welfare. That's the badge around it. We know that looking after animals and disease takes a lot of people's time, and protecting them and treating them and all of those things. So this is about an index to help producers select animals that are easier to manage but are still optimal from a welfare perspective, and the welfare part of it's a very key part of that. So that's about it. I'd just like to really acknowledge MLA for all their support of this work. We could not have done any of this without MLA's funding, and they've funded quite a bit of this work, majority of it. Also like to thank Angus Australia. They've been excellent industry partners. We've got a great relationship with Angus Australia that continues now, and we couldn't do this work without access to their benchmarking animals. So a big shout out, especially to Christian Duff and Nick Butcher, and also to AWI who've funded the recent MLP stuff. Aaron Ingham and Ian Cole, that's my mentors really, which are heavily involved in the work. I won't go through all these people, but there's a whole stack of people we'd like to acknowledge that have helped along the way. So thanks for your time.
Thanks, Brad. Well, I've taken off the photo I was going to ask. Okay, so we're going to have to fly through a few of these. So that first one, immunocompetence equally heritable from sire and dam.
From a non-geneticist, you're probably asking the wrong person.
Yeah, I'm going to say from the data set, you're measuring all that information.
Yes.
So let's just say yes.
Yes, we will say yes.
Excellent. Good. That's one down.
One down.
Okay, so do you, I guess both being ruminants, do you expect immunocompetency response to be the same in sheep as beef?
We see, whether it's the same is a hard question, but we see very similar responses. The antibody responses are similar in magnitude, if that's what the question's about, and the cell responses as well. We just measure the cell response at a different spot. The cattle we measure on the caudal folds on the tail, which is a bit like the old TB tests, if anyone ever saw someone do a TB test in the day. Sheep we do in that wool-free part of the groin, because we tip them upside down on a V-belt handler and test them that way. But they are very similar, I must admit. You see the same spread certainly in responses.
Okay, I'm going to take one more here, and then we'll go to Jock, and then Kim, is yours a question or as well?
Yeah, no, it's a question.
Yep. Okay, so we'll punch through this next one.
The next one's a really good question, and we saw exactly what we would expect. So we get negative correlations with production traits, but the good news is, and this is mainly I can speak about the Angus stuff because we've done a lot of animals, and we've got really good estimates of correlations there. So we do know that immune competence and production traits like growth are negatively correlated, unfavourably correlated, but the correlation is weak, which is good. But it drives home the message for producers: if you're selecting for productivity, you're not selecting for immune competence, and that's exactly what we expected to see. There's lots of information out there, chickens, pigs, dairy cattle. If you select for productivity, you will reduce their ability to fight disease. So that was not at all an unexpected finding, but it's really important that we look at that. So we would never say to people just go and select for immune competence, but what we're saying to producers is continue to select for productivity, but put some emphasis on health and fitness traits as you go, because there are animals out there that are good at both, and we need to be dragging them along together. If we keep pushing hard on production, it will be at our own peril. So,
good message to finish. Okay, I'm going to take one more question. Kim, we'll take yours in the break, if that's okay. Jock?
Brett, did you have an immune did you compare the twins versus singles in the sheep from an immune competency point of view, and if so, was that bias in that table that you showed?
So we certainly haven't looked at it directly, but we certainly fit that into the model. So when we look at an immune competence phenotype, all of those standard things we use for other traits like age of dam, birth type, rearing type, age at measurement, all of those things are fitted in there. But I must admit we've never specifically looked at twins versus singles from that perspective. But whether you're a single or a twin, that effect is fitted in when we generate a phenotype for you. Yeah.
Okay.
If that makes sense.
I'm going to pull up. Sorry.
Brad knows I love the immune competence work, so he's not too worried. I was just thinking, Brad, a lot of the work you're doing is on the young animals, and they have to survive kind of immediate challenges. You know, that's what you're recording in terms of your productivity and stuff. Have you got any thoughts about retesting those same animals when they're mature? Because presumably most of them will live, and see whether they're still ranking the same for their competence, and whether that relates to the colostrum quality, what they put into their calves. But the second thought I had was, if you know they've got lousy immune competence at weaning, is there any treatments you can give them to change it?
Yeah. Good questions. So the repeatability one's a really interesting one, and that's because when you test an immune response in the animal, you can never go back and test the same thing again. The responses are always expected to be different because there's a memory component to it. So repeatability is something we've looked at, and I can tell you that, for instance, from an antibody perspective, secondary and tertiary antibody responses are really quite highly correlated phenotypically, like 0.8 or 0.9. But that's probably not that unexpected, in that if you've got your secondary antibody response, that's your starting point for your tertiary one. So you expect them to be pretty highly correlated. So the repeatability one's a really good question, but it's a really tough one when you're looking at immune responses, because you're never measuring the same thing when you go back again. So it's very different to a weight or measuring whatever, because there's that memory component. Your question about what can we do with the low responder animals, we can protect animals with vaccination. That's part of it, but the problem is those low responder animals may not respond very well to vaccination either. And part of what we're doing now is looking at how our immune competence phenotype relates to responses to general vaccines. So in sheep, we've just collected a whole heap of data looking at what's the correlation between our immune competence measure and response to Barbavax, the Homoncus vaccine, because we're interested to see whether, what we're hoping is with our immune competence stuff, part of that is that we'll help producers have animals that will respond better to vaccination against a multitude of diseases. So the low responders, it's hard to know what to do with those animals, because they're very hard to protect artificially as well, because their immune system just doesn't respond how we'd like it to. So there's two sort of ports of call here. Some people think targeting the high responders is the way to go. I actually think quite the opposite. I think getting rid of the low responders is the way to go. And I think an average animal will generally be protected. There's some level of protection that's out there, and of course the high animals are up here, but a lot of those average animals are protected as well. To me, it's about getting those animals out of the system that aren't protected. So not sure if I've answered your questions, Kim, but the repeatability one is one that we've thought about a lot. It's a very hard one to do when you're looking at immune responses that have got a memory component to them. Plus, we like to test over that stressful period of weaning, which we think is really important. So, it's the best I can do.
Brad, can I just clarify that bit about the response? We know that with any vaccine there are non-responders.
Yep.
You've talked about the repeatability of the low and high. Is non-responding in that same thing? You go and vaccinate them again and they don't respond again completely?
Yeah, so what we see, and we've gone back and tested animals over and over again in the past, and what I see is a high responder animal just reaches that protective level quicker.
some of those animals that aren't quite as high responders will eventually get there, but you've got to keep priming them to get them there. But there's some animals also that never get there. So you get the whole range. You get those high responders which might only take, 2 vaccinations to reach this antibody level. An average responder might take 4 to get to the same level of protection. And there's some animals that just never quite get there, so they're never protected. But protection's very different to looking at antibody response. So when we measure an antibody response in the animal, the same antibody response doesn't always equal the same protection in 2 different individuals. So it's a very difficult question to answer, and we've got to be really careful to separate our measures of antibody and protection, because we're not measuring protection, we're just measuring how strong their immune response is. And it's by looking at health outcomes that we're trying to actually relate that to how they handle disease challenge. The only other way to do it is to actually challenge the animals with a disease, and we don't want to go down that track.
Yeah, cheers, Brad, for that presentation. I was just wondering, do you actually have some solid genetic markers for the immune competence now? Yeah,
so interesting you asked that question. So the work we've done with Angus, we've actually done a GWAS on that, and it was really interesting that there's a couple of really strong markers coming out for antibody and cell, and that was a little bit unexpected because, I've heard numbers like 7% of the genes are actually related to immune function. So to find single genes that have major influence was probably a bit unexpected. So we're just in the process of trying to look at the biological function of those genes, but there's certainly a couple of markers, one for antibody and one that's cell, that are right off the chart when we do a GWAS on these animals. So it'll be, and the nice thing is they actually make sense when you look at what they are, but we're looking at the biological function of those genes, and that'll be interesting for responses to vaccination, both in livestock and in humans actually, is what we're interested in, because we get vaccinated against tetanus as well. So yeah.
One last question, yours, and then.
I just have a question about, so if you're measuring like the trait WEC, so worm egg count, is that inherently selecting for a decrease in susceptibility, an increase in susceptibility to viruses?
No,
and is there a trait for viruses? Or?
So I can tell you, in dairy cattle, it's been pretty well shown that there's a negative genetic correlation between your ability to respond from an antibody to a cell perspective. So they're negatively correlated. In Angus cattle, we don't see that. They're positively correlated, but it's weak. So it's a little bit different story from what we see in Angus cattle. But I guess my message was not, if you're selecting for resistance to internal parasites, is not going to necessarily make those animals more susceptible to a virus, but it depends how hard you push down that track, if that makes sense. But there still will be animals that are highly resistant to worms that are also highly resistant to viral infection as well. But you can't take them as being the same thing, is I guess my message. Don't assume that an animal that's really highly resistant to WEC will also be highly resistant to a viral infection. It won't mean it's susceptible, but it doesn't mean it's resistant. So selecting for one disease doesn't mean you're making an animal that's resistant to lots of other diseases. That's, I guess, my message, rather than that they'll be actually negatively correlated. Yep.
Okay,
I was
ready?
just going to say that in pigs within full SIBs, you actually get really big differences. So just because they're littermates doesn't mean they'll have the same immune competence.
Excellent.
Thanks, Kim.
Okay, thank you. So I think in terms of just wrapping up there, obviously a very interesting talk about a future area of measurement or traits that we as an industry should definitely think about. So why don't we give our hands up for Brad? Very interesting and thought-provoking presentation about something that, potentially may not be we're directly measuring ourselves, but is also available moving forward.